Okay, can you guys see it? Okay. So today we're going to be talking about cellular metabolism, which is kind of how your cells eat. So this is going to be the last lecture of this course. So it's really, really great to meet all of you, and yeah. So I'm just kind of going to go through this, and then if you have any general questions, then you can ask me at the end, or if you have any questions pertaining to this, then ask it throughout. So yeah. Okay, so cellular metabolism. You guys probably know how you breathe. Oxygen comes in, carbon dioxide goes out. So that has to do with your cellular metabolism, which is known as cellular respiration. So you breathing is normal respiration. Cellular respiration is the breathing of your cells, in a sense. So the way this works is within your cells, you have organelles called mitochondria, which we've covered a little bit. So mitochondria is the site of the full process of cellular respiration. Part of it occurs outside of the mitochondria, part of it occurs inside, but for the sake of it, mitochondria are the organelles that cellular respiration pertains to. So here's just an overview of what happens, and as you can see, it's really complex. There's a lot of moving parts, but they all work together pretty seamlessly, and it makes it pretty unique. Okay, so sorry for the blur, but this is the chemical equation of cellular respiration. Cellular respiration is a chemical reaction, and because of that, it is subject to a specific equation. This equation relates the reactants or the substances kind of changing to the products, which are, well, the products. So your reactants are sugar and oxygen. The molecular formula for sugar is C, so it's C6H12O6. I don't know if we covered this, but if any of you guys know what that means, like what elements are in it and how many, then could you type that into the chat? Anyone? Or you can unmute yourself. Yeah, no worries. So basically what it means is you have 6 carbon atoms, 6 hydrogen atoms, and 6 oxygen. 12 hydrogen atoms and 6 oxygen atoms. So the subscript kind of tells you how many atoms you have, whereas the letter is the letter of the element itself. So that's the formula for sugar as a molecule. But anyway, sugar reacts with 6 oxygen. So this O2 is oxygen gas. There are 2 atoms in oxygen gas, and these 2 substances react together to form 6 carbon dioxide molecules, 6 H2O molecules, and ATP. So do you guys remember what ATP is? ATP is your cell's energy. Your cells use ATP to carry out all of their functions, and it's really a vital part of, energy. It's a vital part of your cellular processes. Okay. So the first part of cellular respiration is glycolysis. Glycolysis is generally the first step, and it occurs outside of the mitochondria within the cytoplasm. The reason I say generally the first step is because sometimes it's the only step, if that makes sense. But yeah, it occurs outside the mitochondria within the cytoplasm. So you can imagine that for a cell that doesn't have mitochondria, say a prokaryote, then they can still carry out glycolysis. It is the initial breakdown of the sugar, and it creates pyruvate, NADH, and ATP. It does not require oxygen and is a form of anaerobic respiration, which ties into what I mentioned earlier about prokaryotes. So if you were to have a prokaryote at the very bottom of the ocean, they could still carry out glycolysis and they could still generate ATP. The way they do that is through glycolysis, and then it can go to another process, or they can just stop after glycolysis. There's a lot of different ways. And then the things here that we should talk about are, we already went over ATP, but NADH is an electron carrier. And basically what it is a molecule that can accept an electron and also give an electron. So what NADH does, and there's another molecule called FADH2 that we'll cover later, but what NADH does is it accepts a molecule, it accepts an electron, and then it'll donate that electron at the very end of cellular respiration. And then pyruvate is kind of just a broken down version of the sugar, glucose. Pyruvate, one glucose molecule will yield two pyruvate molecules. So yeah, okay. So this diagram kind of shows you the steps of glycolysis. So you start off with your glucose molecule, and glycolysis actually requires ATP to occur. So let's say you had a cell that had no ATP, it wouldn't be able to generate the ATP because you need some to make some. So glycolysis, your glucose molecule will accept 2 ATP and it'll yield 2 ADP. So it's basically taking the energy from ATP and giving you ADP outside. And then that is used to split it into 2 pyruvates. And then it'll generate 4 ATP here. Can you guys see my cursor by any chance? I feel like there's a better way to do this. Oh, you can. Okay, great. So 4 ATP is what will be generated out of it, as well as 4 NADH. So basically, they're giving 4 electrons to 4 electron transport carriers. So the net gain is 2 ATP because you used 2 and you got 4, so 4 minus 2, as well as 2 molecules of pyruvic acid. Okay. So here are the net results just summed up for you guys, and this is per single molecule of sugar. 2 pyruvate, 2 ATP, and 2 NADH. Okay, so here we have fermentation. Fermentation is it's not cellular respiration. It's different. It generally does not happen in eukaryotes. It doesn't happen in plants. In humans, let's say you're not getting enough oxygen, then usually like your arm muscles, your biceps, your muscle cells will undergo fermentation because they need more oxygen in that area and they're just not getting it from cellular respiration or to be used in cellular respiration. Okay, so what is fermentation? It is an anaerobic process that allows organisms to produce energy in the absence of oxygen. It follows glycolysis, and there are 3 main types. You have lactic acid fermentation, which is probably the most common that you'll definitely learn about in a biology class. It's really common in yeasts. Then you have ethanol or alcohol fermentation. That's kind of like how you make beer and wine. And then there's acetic acid fermentation, which is probably the least common. It's kind of like an extra step. Okay, so here's lactic acid fermentation. You have the 2 pyruvic acid molecules as products of glycolysis. Then 2 NADH molecules are taken in and 2 NAD plus molecules are left. And this yields 2 lactic acids. So what this does is the lactic acid is kind of just a byproduct. Your real energy does come from the glycolysis reaction. So then here we have ethanol or alcohol fermentation. So once again, the 2 pyruvic acid, it releases 2 carbon dioxide molecules and forms ethanol, as well as taking in 2 NADH molecules and releasing NAD plus. So once again, all of the energy comes from glycolysis. And then here's acetic acid, which is kind of an offshoot of the last one we looked at. Basically, once you have your ethanol molecule, a reaction occurs and it forms acetaldehyde, and then it forms acetic acid. This one is not super important at all. Okay, do you guys have any questions thus far? then let's keep going. So the link reaction is kind of special. It's not the second step. It's actually like step 1.5. So it is the intermediate minor step that occurs between glycolysis and the Krebs cycle. It is the transformation of pyruvate into acetyl cofactor A. Acetyl cofactor A is what's used in the second step. So this transformation is crucial. You can't go on unless the link reaction occurs, and it occurs in the matrix of the mitochondria. So this is where our reaction moves from the cytosol to the mitochondria. Okay, so this is how it works. You have your pyruvate molecule, and there are a whole bunch of things that happen here. It takes in cofactor A, all of this, it releases NADH. But the ultimate important part is that it releases carbon dioxide and acetyl cofactor A. So acetyl cofactor A is kind of a combination of acetyl and the cofactor A, and you'll see how they're kind of disjoint in the next step. But the important part is that here is our first time that we're releasing carbon dioxide, and you'll see that carry through, especially in the next part. So your net results are 2 acetyl cofactor A's, 2 NADHs, and 2 carbon dioxides. And once again, this is per molecule of sugar, so per 2 pyruvate. So here we have the Krebs cycle. The Krebs cycle is super, super intense, and if you were to take any organic chemistry class, this would be the bane of your existence. It's mega intense. Luckily, in high school, at least in AP biology, we didn't have to know all of the ins and outs of the Krebs cycle. You just need a general overview. So what is the Krebs cycle? It is the second step of cellular respiration. It's also known as the citric acid cycle or the tricarboxylic acid cycle. It is an extremely chemical process that involves a lot of bond breakage and formation, which is why it's super difficult in OChem. But the acetyl group in acetyl-CoA binds to another molecule. The CoA coenzyme A, it's also known as cofactor, is used to catalyze more link reactions. And this new molecule is what completes the Krebs cycle. So here is just kind of the first, this is kind of, it gives you an idea of what's released. It doesn't really tell you what happens, but if you'll notice, these numbers are decreasing, and the C refers to carbon. So acetyl-CoA goes into the Krebs cycle, and it is originally a 6-carbon molecule. Then something happens, and now it's a 5-carbon molecule, once again, 4-carbon molecule, once again. Well, it does this a couple times. But what you'll notice is that each time the number of carbons decreases, it's because, what do you call it? Carbon dioxide is released. So the Krebs cycle is the major site of carbon dioxide release, as well as it creates a whole bunch of charged electron transporters. So here we have NADH, here we have FADH, and more NADH. Okay. So the net results of the Krebs cycle are 2 ATP, it takes in 2 and releases 4. 4 cofactor 2, or oh my gosh, 4 carbon dioxides, 2 FADH2s, and 6 NADHs. Okay. Do you guys have any questions thus far? Okay, then I'm gonna keep going. Here's our last step. Our last step is oxidative phosphorylation. Oxidative phosphorylation is composed of 2 main sections, the electron transport chain and chemiosmosis. The ETC, which is the electron transport chain, processes electrons from all those NADH and FADH2 carriers, and it uses these electrons to create a proton gradient. A proton gradient is basically what happens when you have a higher charge on one end versus the other end of a membrane. So in this case, we're talking about the membrane of a mitochondria. And what happens is on the outside, the area is very positive because of all the protons, whereas the inside is relatively negative. So the way that not just biology and chemistry, but like the world in general works, is it favors equilibrium. And equilibrium in terms of like concentration, but also equilibrium in terms of charge. So it wants the same charge on either side of the membrane, and the oxidative phosphorylation, specifically chemiosmosis, will take advantage of this desire for equilibrium. So that's how the proton gradient facilitates the generation of many ATP molecules in chemiosmosis, and it is also the first time oxygen is required, the first and only time. So here is a super detailed diagram of your electron transport chain. So right here is your intermembrane space. Here's the outer membrane and the cytoplasm. Here's the inner membrane and the intracellular matrix, basically like the inside of the mitochondria. These huge white spots are your proteins. They're called membrane proteins or electron accepting proteins, I guess in this case. This line is kind of the pathway the electrons would take through the membrane and through the proteins. E minus denotes electron, and H plus in this case refers to protons. So you'll have your NADH molecule, and it'll deposit the electron in the first membrane protein, and it'll emerge as NAD plus, ready to be recycled. FADH2 will do the same exact thing. The electrons will travel through each protein, and as they're doing so, these H plus molecules are able to be pumped outside. And then at the very end, we have chemiosmosis, which is this. And you have your H plus molecules, and they funnel through this, and as they come down through the membrane, they catalyze the formation of ATP. So basically, the movement of H plus ions across the membrane creates like potential, it creates energy, potential energy, and that potential energy is used to join ADP to a phosphate. This contraption, it's really a protein, is known as ATP synthase. ASE because it's an enzyme and it catalyzes the joining of the two molecules. Okay, so here's like a more visual, I guess, image. But yeah, it's basically the same thing. The important part that we didn't cover in the last photo is this right here. So you have your O2 molecule, and you have all of these H plus ions that are coming back down. And the way that these electrons are drawn through the electron transport chain is by the presence of the O2 at the end. So as you guys know, H2O is water. That's why we have the formation of water. And what happens is the H plus ions want to join with this O2 gas. The O2 gas will split. It's a very chemical process. But what happens is the O2 gas will split and join with two hydrogen or H plus ions. The problem is this isn't a hydrogen, it's an H plus ion. To make it a hydrogen molecule and to form H2O, the O2 and the H molecules atoms need electrons. And because we talked, I think briefly we touched on it about electronegativity, oxygen is a very, very electronegative molecule or element, meaning that it can draw electrons a lot more readily. And so it basically just pulls them through this transport chain, and it's really the driving force behind everything that's happening here. Okay, so here's just like a quick summary of the last part. So it starts off with NADH and FADH2 transferring electrons to membrane proteins on the electron transport chain. These electrons move from high to low energy as they move through proteins. This energy is used to pump H plus ions across the membrane. The O2 oxygen gas molecule takes electrons, splits, and binds to H plus ions, and this is what creates H2O. And as this happens, the H plus ions travel down the proton gradient and back into the matrix through ATP synthase. ATP synthase pulls energy from the H plus movement to bind a phosphate group to ATP, or this is supposed to say ADP to form ATP. And here we have our net results. So this is kind of, this isn't what happens from all cellular respiration. This is just oxidative phosphorylation, so that ETC and ATP synthase. So you will get, 34 molecules of ATP, 12 molecules of H2O water, 2 FADH, and 10 NAD+. So if you remember, FADH was denoted as FADH2 while it had an electron. Now it's just FADH. NAD plus or NAD plus was denoted as NADH while it had an electron. Once it gives it up, it's now NAD plus. The reason for that is now it's got a positive charge on it and there's no more hydrogen. Okay. Do you guys have any questions for me? This is the end of the presentation. So if you have any questions on this or on the course in general, please let me know. I'll give it a few minutes.